Very good evening to everyone, and good morning to Dr. Mandal and colleagues and friends who have joined from US for this wonderful session on COVID-19 vaccines and variants. Times to get back to normalcy. Yes, we are looking for that. I think all the countries are looking for that, and we have to get back to normalcy. So this is this has come at the right time, and we are going to go ahead to listen to Dr. Mandal about what is his view about the different vaccines and the variants. So at this juncture, I welcome our today's speaker, Dr. Manas Mandal, who has graciously accepted our invitation and will be giving us the insights of today's topic. I welcome you, sir.
Thank you, Dr. Wadhwan.
Yeah, I do welcome our Chief Executive Officer of JSS Academy of Higher Education and Research, Mauritius, Dr. Praveen Mahadev.
Thank you.
I welcome Mr. Naveen KP, the Registrar of JSS HER Mauritius. Leadership from the parent institution, JSS Academy of Higher Education and Research, Mysore, Dr. B. Suresh, and other Dr. Vishal Gupta and other leaders from Mysore who have joined us. I welcome the coordinator, Dr. Khyati Modgil, and
thank you.
Other faculty members, colleagues from JSS institutions and the other institutions as well. Last but not the least, I welcome all the participants. We are made to understand that we have a good number of registration, and slowly you can see the people are joining. So I welcome all the participants for attending this particular session. So with this, I request the Chief Executive Officer, Dr. Mahadev, to give us the opening remarks for this webinar. Over to you, sir.
Thank you, Dr. Ashish. Distinguished participants, colleagues, ladies and gentlemen, good morning, good afternoon, and good evening, depending on where you are. This is the beauty of technology. I'm also very pleased to welcome you all to this webinar on COVID-19 vaccines and variants. Despite the fact that the registration was not compulsory, we note that we have more than 900 registered participants. We can see the numbers increasing on the counter in this virtual conference room, coming from various countries across the globe, including Mauritius, India, USA, UK, Malaysia, Korea, Oman, Iraq, Algeria, Singapore, Sudan, and Philippines. Ladies and gentlemen, 2020 has passed, and here we are in 2021. None of us will ever forget these past 2 years because of COVID-19. The COVID-19 pandemic now threatens the lives and livelihoods of every citizen globally. It has sparked a slew of anxieties, including health, agony, and bereavement. Everything is affected. Our everyday life, the state of our economies, the future, education, jobs, careers, cross-border travels, supply chains, food consumption, healthcare, and even corruption and abuse, drug abuse. While health professionals were figuring out how to treat the virus, researchers in the pharmaceutical sector set out to investigate existing drugs that might help treat patients with COVID-19, potential new medicines and drugs that would help mitigate the symptoms and long-term effects, and the production of vaccine to protect us. The vaccine, or I should say vaccines, came early this year and was good news for all of us. But with the vaccines came a number of questions in everyone's mind. Which vaccine? How long it will be effective? Dose intervals, safety, efficacy, etc., etc. Whilst we were having these questions in our minds, the virus acted smartly and mutated itself to give different variants, creating more confusion and more questions. With all these questions and confusion, we at JSS Academy of Higher Education, Mauritius, thought it appropriate to provide some answers and clarifications on the matter. We have therefore requested Dr. Manas Mandal, an expert in the field, to enlighten us and give us more details and information about the vaccination and the different variants, the vaccines, and protection against the virus. I'm very grateful to him, and I thank him for having accepted our invitation. Hopefully, after his lecture, some doubts will be dissipated, and we will learn to live with this virus and to move back to normalcy, or what we call the new normal. With these few words, I will now request Dr. Ashish Wadhwani, this evening's convener, to introduce Dr. Mandal, who will thereafter make his presentation on the topic of the day, COVID-19 vaccines and variants, time to get back to normalcy. I wish you all well, keep safe, and thank you for your attention.
Thank you very much, Dr. Mauritius, for setting up the right tone. It is my proud privilege to once again welcome Dr. Mandal. To brief about Dr. Mandal has completed his MSc in Physiology in 1985 and PhD in 1992 from Jadavpur University, Calcutta. And then he moved to USA. He worked as a research fellow in Department of Microbiology and Immunology at Chicago University. Then postdoctorate fellow again at the University of Chicago between 1994 to 1997, and moved to University of Michigan for one more postdoctorate fellowship in Department of Pharmaceutical Sciences, College of Pharmacy, University of Michigan from between 1997 to 2001. Currently, Dr. Mandal is the Fulbright specialist and the professor of Pharmaceutical Sciences, Roseman University of Health Science, College of Pharmacy. He has worked at various capacities and vast experience of 30 years he carries, and he will be talking to us about the COVID-19 vaccines and variants. He's, to make a mention that he is the Fulbright specialist in the US Department of States of Education and Cultural Affairs and World Leadership at the World Learning position from August 2019 to August 2022. He has various years of experience and worked at the various capacities at different institutions in USA. He is the member of all top pharmacy bodies, to name a few: American Association of Pharmaceutical Sciences, American Association of Colleges of Pharmacy, American Society of Microbiology, American Association of Advancement of Science. He has 70 plus research papers to his credit at various journals, and chaired various scientific sessions. With this brief, I introduce to you all Dr. Mandal, and we are eager to listen to your thoughtfulness about the COVID-19 vaccines and variants. Over to you, Dr. Mandal.
Thank you, Dr. Wadhwani. That was so kind and so long introduction. You never needed to do that. Yes, by training, I'm an immunologist and vaccine researcher. I worked at the University of Chicago, then University of Michigan, looking into the viral vaccine development, where we wanted to target a particular pathway of antigen presentation triggering virus antigen specific vaccine development. And I also thank Dr. Mahadev, Dr. Suresh, Dr. Mudgil, and all the other members of JSS at Mauritius and in India, Mysore. It's my privilege to be back with this platform, this august body of scientists and educators. And I really thank you all for giving me the opportunity to talk to a larger audience, not only to the people of Mauritius and other countries. There were 7 or 8 other countries named, and including US and India. What I'm gonna do today is to develop a sort of a picture about this infection. Since 2020, we have been through this pandemic, and I personally believe perhaps we are at the very end of a long, dark tunnel. We can see the light, but there are precautions, there are cautions needed to be taken. We want to understand about this infection, various vaccine development approaches, and various vaccines that are currently available in the market worldwide. With that short introduction, let me go begin this presentation. Give me a minute to open up the presentations. Now, one of you need to tell me that, do you see my presentation? So with this screen, if it's reasonably visible to your end, I will begin my talk. So today we are going to talk about the vaccines and variants for COVID-19, and is it time to get back to the normalcy? These are the learning objectives. We'll talk about infection and pathophysiology, and then go into vaccine development approaches and current vaccination that are administered worldwide. After that, we'll talk about vaccine safety and efficacy, meaning these are very new vaccines. There were lots of concerns about their safety and efficacy. On the heel of that thought, we will talk about the recently emerging variants. There are lots of concerns and questions whether the vaccine that those have been generated against the original infections genetic sequences, will that vaccine still protective against the variants? And at the end, we will discuss how do we get back to normal or normalcy from this point. I'll begin with a quote from The Plague by Albert Camus. It talks about this. Yes.
Can we have it in the slide show mode?
Slide show.
It will be good.
Okay.
Yeah. Sorry to bother you. Yeah, perfect.
No, it's all right. Is that better visible from your end? No? Do you see it now? Okay. So now you have to little bit guide me. Do you see me looking at the slide presentation, or I'm turning away? Because I have two screens, both are showing up as a presenting screen.
Yeah.
Is that okay now?
Yeah, it's fine. It's fine. We are at the first slide only.
You are at the first slide. Okay. So let me move the camera now. So let's talk about the variants and vaccines. Now, I'm sorry about that. It's a technology. It works, but sometimes you have to work through it. So this is the description of plague by Albert Camus. In 1947, he writes, everybody knows that pestilences have a way of recurring in the world. And currently we have a pestilence on hand. It ends, there have been as many plagues as wars in history, yet always plagues and wars takes people equally by surprise. We were surprised. At the beginning of 2020, this is the beginning, on January 9, 2020, a mysterious infection was developing in the Wuhan province of China. Subsequently, it came to known as coronavirus infectious disease 2019 because it started appearing or happening in 2019. In short, that is COVID-19. Very recently, WHO reports shows that most likely COVID-19 pandemic originated and zeroed in the animal markets, not in the labs. That was reported by WHO, and this is an excerpt from the Nature that published the full-fledged study 2021. And for all the presentations or the data point that I will show you today, I have the relevant references. If you feel like, you can go back to the original article, original source of information to read yourself. Because what we learned collectively through this pandemic, through this infection, there are lots of mis and disinformations that goes around, and we have to combat the mis and disinformation with correct sources of information. So what is coronavirus infection? It's a family member of coronavirus. We have seen this family member before, such as Middle Eastern respiratory syncytial virus, CoV, and SARS-CoV, which also occurred or infected in Far East, such as Hong Kong. It's a single-stranded RNA virus of about 80 to 220 nanometer in size, very distinctive, and carries a large 20 nanometer long club-shaped spike that you see on the right upper hand side of this diagram of the coronavirus. And these spikes are famously known as spike protein that binds to the H2 receptor structures expressed all over our body, including the lung, nasal epithelial cells, and nasopharyngeal passages. The H2 receptor was not expressed to bind to SARS-CoV-2 spike protein. H2 receptor is involved in blood pressure regulation and cardiac functionality in our body. It has a physiological purpose. But this virus almost hijacked the usage of this receptor structure that expresses all over our body and somewhat at the skewed expression level in the lung, nasal passages, and epithelial cells. A specific proteolytic enzyme such as TMPRSS2 primes S protein after S protein bound to the S2 receptor structures. And at that point, this virus gains entry into an individual, setting up the infection. And the skewed expression level of the S2 receptors in the upper airway may be correlated with enhanced transmissivity and virus reservoir. There is a very important information in this statement, which is this is an airborne infection. Also, this observation led to the development of the nasal swab, RT-PCR testing, and other testing. This slide shows the SARS-CoV-2 entry and life cycle. On the left-hand side, we have original SARS-CoV, then current SARS-CoV-2. This blue diagram is the current infection. And many scientists believe that it originated in the bats, subsequently gotten into the animal, and when that infected animal was being handled in the wet market in Wuhan province, then someone, the individual who was handling that animal, got infected with the virus. This is called zoonosis. Originally, we are not the carrier of these viruses, rather than these viruses exist in the nature. But upon zoonosis, we remain the reservoir of this virus infection, and we transmit the virus. On the right-hand side panel, it elaborately shows this human coronavirus being bound by the S2 receptor structure, and subsequently, it is taken inside the phagosomal compartment of the infected cell, where the viral RNA will be released, and through the translation and transcription through several steps, this virus will generate its structural proteins such as nucleocapsid protein, M membrane protein, E envelope, and most famously spike protein. After that, this viral particulate matter will be repackaged into the new variant, and it will be released to the adjoining cells to set up the infection, or at that point, if the infected individual is in close proximity with another individual who hitherto is uninfected, then that person can catch the viral particle released airborne through sneezing, coughing, and even regular talking. We all release aerosols when we speak. Now, if an infected individual speaks in close proximity with another uninfected individual who is not wearing a mask or physical barrier or not at a distance, then that individual might as well catch the infection. Down below, it shows that these structural aspects of this viral infection are the drug targets for the vaccine production, such as S1 and S2 domains of the S proteins and various other monoclonal antibodies that are commercially available currently. They target various epitopes of the S protein specific antigenic structures on the viruses. With that, I will now focus our attention to the newly generated various mutations and mutants that are circulating all over the world. Viruses do mutate. This is nothing new for COVID-19, because we have to remember that viruses are inert particles, meaning they do not replicate, cannot replicate without getting entry into a live system. Many mutations in the S protein have occurred very early on in 2020, D614G, D2G, that mutation, single amino acid mutations, and N501 and H69V7, all these mutations have been discovered and described as of 2020, early 2020. And at this point, it is estimated that about 2 mutations or more are occurring per month. Notable mutations or cluster of mutations led to the designation of variants. What are these variants? Most famous variant is B117, originally generated in Kent, UK. Then we have the Africa-specific variant, which is B135. P1 is Brazilian variant, and most recently B.1.617.2 India-specific variant. Now, there is a consensus opinion developed in last several months that we should not call these mutants naming the source of country or origin of the country where it originally originated, because viruses do not have any geographical boundary or they do not need passport. Essentially, they do only mutate when they spread the infection, and that infection can be spread in US, in UK, in India, in Brazil, or anywhere in the world. So it creates a stigma in the people's mind when we name a mutation or a mutant or a variant as per the name of the country. Therefore, scientists and clinicians worldwide agreed on a common naming of these mutants or variants, such as alpha variant for UK specific variant, beta for South African or Africa variant, gamma for Brazilian, and delta for recently discovered or identified India specific variant. What did these variants do? Essentially, they do increase the transmissibility, may trigger increased pathogenesis and severity of the disease. Particularly both alpha and delta variants showed increased infectivity and disease severity and hospitalization compared to the original strain of the infection. And the right-hand side diagram shows the these red dots are the spike proteins, various amino acid sequences where these mutations are being located and is occurring. And in the top, this ribbon diagram shows the ACE2 receptor structures. That's binding to the receptor binding domain of the spike protein. And that's the way the virus binds to the ACE2 receptor structures and gains entry. Now, with these mutations, there are some concerns. And not only for the increased transmissibility and pathogenesis, but many mutations may compromise ACE protein specific vaccine effectiveness and immune protection. I will cover that aspect at the very end of my talk, but currently let's agree that these are the variants of concerns, all these mutations. And this slide essentially shows that viruses only can mutate when they are infecting an individual, and original virus infection, then subsequently it will replicate, and upon replication within the infected individual, it might gain a mutation in one of the amino acid sequences in spike protein and many other structural proteins in the virus. Subsequently, this infected individual now carrying the mutation may transmit the infection mutated version into other individuals. Thus, we got the UK specific Kent variant, South African variant, and Brazil variant. And this is a few months old data, but essentially we can add India-specific Delta variant below this slide. Let's understand this infectivity and mortality of COVID-19 a little bit before we move forward into various vaccine development approaches. Because when a scientist thinks about developing a vaccine against an infection, they first wanted to understand how and what is the infectivity, how it infects, what is the pathophysiology of this infection. Unless we understand the pathophysiology of the infection, a proper vaccine cannot be developed, meaning vaccines should be designed to not only prevent infection and thus infection-induced pathogenicity or clinical illness. Thus, let's a little bit talk about infectivity and mortality that is observed through COVID-19. It's a very high infective virus and triggers high mortality, about 1%, and a close comparison means a comparative estimate will be 0.1% of mortality that is seen with seasonal flu influenza virus infection. Therefore, compared to seasonal influenza, COVID-19 comes up with a 10 times more mortality. Infection fatality ratio, IFR, also differs. Differs means depends on the country to country and variant to variant specific infection. About 1.15 in high income nation versus 0.23% in low income nation. There are reasons for that. It's not very directly easy to understand, but in high income nation, it has been observed that people live longer. Many individuals live in a long-term care facility, nursing home facility, etc. Therefore, as the collective age in a high-income Western country is higher compared to rather low-income nation, fatality rate is also higher in Western world high-income countries compared to low-income nations. That's the way we want to understand that. One more important aspect of this virus infection and its infectivity is R0 value, which is the reproductive ratio, meaning from one infected individual, how many other individuals can get infection, which is currently calculated between 2 to 2.5, but this number also varies depending on the region of the country where this infection is taking place. What it states that one infected individual will at least release the infection and be part of the infecting at least 2 to 3 individuals, and it will proportionally increase. Fatal disease is highest in people over 65 of age, deadlier for older people, 0.1%, less than 40-year-old. It goes up to 5% over when the age goes up 80 years and above. Asymptomatic infection prevalence above 45%, and asymptomatic patients can have 75% transmissibility, meaning if I am infected, I'm asymptomatic carrier of the infection. I do not know that I got the infection in me, yet I can transmit the infection longer than 14 days. And interestingly, about 50% of the COVID-19 transmission do occur before one gets sick. With that infection information and mortality information, let's get into the clinical feature, what it does upon infection. It disproportionately infects males over 50 years of age. Comorbidities, obesity is the number one, with high blood pressure, cholesterol, diabetes, heart and kidney disease. The most clinical manifestations of the disease in the early phase, fever, chill, cough, subsequently fatigue, muscle, body aches, loss of taste, smell. In the mid through later phase of the infection, may develop shortness of the breath and nausea, vomiting, diarrhea. Most severe disease condition is respiratory distress syndrome, acute respiratory distress syndrome, ERS. If that develops and oxygen saturation goes below 92%, then we need oxygenation, ventilation, etc. In the white blood cell compartment, we see leukocytosis and lymphopenia, meaning white blood cell exhaustion is a profound biomarker, cellular marker for this infection. What it tells us that white blood cells that fights viral infection, any viral infection in our body, with this infection, these white blood cells, particularly CD4 positive T helper cells, they get exhausted. They just can't cope up with this virus infection anymore. Thus, they get exhausted and they die out in a large number. Neutrophils also suffers the same fate. They get exhausted. And neutrophils are the first sentinel for clearing up of any infection, including this virus infection in our body. So we can imagine what is the magnitude of this infection. It is quite different from any other virus infection that we have ever seen in us. Biochemical markers, ALT, liver function enzyme test, ALT, LDH, cell death marker, D-dimer, clotting cascade specific biomarker, and various inflammatory biomarkers such as C-reactive protein and ferritin have been found significantly higher in severe cases compared to the moderate cases. And pro-inflammatory cytokines in these severely infected individuals, such as interleukin-1 beta, IL-6, TNF-alpha, and IL-10, significantly higher in those individuals. And it's also markedly high also in moderate infected individuals. Taken together, this is an overall profile of a COVID-19 diseased individual. Typical presentations, once again, fever, dry cough, exhaustion, myalgia, maybe shortness of breath, less frequently nausea, diarrhea, headache, and cutaneous various rash type of manifestation and swelled toes. And there has been multi-inflammatory system in causing infection described in the children, which is in short known as MISC. It resembles Kawasaki's disease, not quite well understood, but there are some hints. Probably these children are not capable of triggering very early on innate immunity through interferon type of responses. When that happens, then they could not clear up the viral infection and they go into the hyperinflammatory disease condition. And most severe form of this disease is once again acute respiratory distress syndrome, ARDS. And here is the right-hand panel. You see the enlarged toe, clotting cascade defects. And in the middle panel, you see the lung quite infiltrated with the white blood cells. That's the histopathology see. And this is a glass-like opacity in the infected individual's lung, maybe having a huge amount of secondary infection, pneumonia, and also SARS-CoV-2 COVID-19 infection. Infection timeline and kinetics, this is very important to understand, not only to have a proper detection, also to come up with a vaccine development protocol triggering immunity, meaning IgG or other classes of antibody responses, and coming up with a treatment protocol. This virus can incubate in someone about -10 days through -5 days, meaning if on day 0, someone showing the onset signs of infection, that individual perhaps gotten infected with the virus or carrying the virus in them could be 5 days before through 10 days before the first sign of infection when shows up in that individual. From that point, we can take a nasopharyngeal swab to determine the viral load and detection through RT-PCR, which is doable pretty high level until about 1 week upon first signs of infection. And initially there will be fever, subsequently there will be pneumonia, and if the pneumonia and lung infection goes out of hand, maybe it requires mechanical ventilation. About day 3 onwards, the infected individual will start making antibody responses towards the virus infection. And those antibodies will be IgM class of antibody to begin with. Subsequently, about day 7 onwards, there will be high level of IgG class of antibody. Now, once the antibody is generated within a week and afterwards, we can take a little blood sample and send it to the lab to determine the antibody responses or detection of SARS-CoV-2 specific antibody in that infected individual. But prior to that, we got to depend on the RT-PCR to diagnose and detect the infection in this individual in this phase of infection. Subsequently, even with the antibody responses, a person's viral load doesn't go away very easily, and that person unfortunately may end up in mechanical ventilation. And that can go up to 3 to 4 weeks or even longer. Translating that information, we can have a viral phase, then cytokine storm phase, meaning where inflammatory cytokines are very high in the infected individual's lung and systemically. And that actually causes the most devastating organ damaging effect in the infected individual. Where do we see it? We see it more severe infected individuals compared to the moderate or low level of virus infected individuals. And following that cytokine storm, then there could be coagulation phase and late hyperinflammatory phase in many of these individuals. So this is the kinetics of the infection or infective. And with that kinetics, once again, it shows that what are here is the patient being admitted and lung, maybe neurological involvement, cardiac involvement, and in the circulatory system, kidney can get impacted. And by week 4, usually the viral will be cleared from the system and it will either be undetectable at a very low level of infection. That's a normal scenario and that's a good outcome. But there has been a phenomenon described as COVID long haul or long hauler who carries the virus in them, perhaps at a very, very undetectable level. It's not very well understood, but those viral infection, primary infection mediated symptoms, some of them exists for a long time, for several months in the infected but so-called recovered individual. What's that long haul COVID manifestations? PTSD, anxiety, depression, sleep disturbance, cognitive disturbances, fatigue, muscular weakness, sometimes clotting cascade defects and chronic kidney disease. And that can persist up to 6 months. We do not understand these long haul COVID manifestations, but essentially it's a persistent hyperinflammatory states and up to 30% or more individual who shows the long haul COVID syndrome can have cognitive or neurological disturbances, including shortness of breath. With these clinical manifestations, we need a team of physicians, clinicians to manage a patient. And that is the very hallmark of the disease management. And unfortunately, worldwide, this is not easily available because we need a team of various specialists, such as pulmonary cardiovascular specialists, that will take care of the breathing difficulties, then hematologists, neuropsychiatrists to treat for the neurological syndrome, PTSD, etc. Then nephrologists, renal, and primary care physician at the end, meaning after the discharge, rehabilitation of the patient, patient education, and etc. And this is very important in the management of a COVID-19 patient. Again, this is not easily available, but this is the ideal way we should look at this infection. It's just not another garden variety of virus infection, which is cleared easily through our white blood cell activation or through some antiretroviral drug usage. We have to understand this very clearly, that it requires a team of physicians and specialist clinicians to manage a COVID-19 patient who requires hospitalization, who requires long-term care. So with that background, let's talk about what type of immune responses we trigger when we get infected with COVID-19, means coronavirus disease 2019. I would not go to the very nitty-gritty detail that is shown in this slide or the diagram, but you can read yourself in Nature Review Immunology. This is a very good article about regular virus infection and what happens in terms of any virus infection that infects us, what type of immune responses in the compartment of innate immunity. When we get infected, any virus infection or bacterial infection, there are two types of immune responses we do trigger. Either it will begin with innate immune responses, then subsequently adaptive immunity will kick in. For clearances of virus infection, interferon alpha and interferon beta is the signature innate immune responses, because we naturally release these interferons, alpha and beta, type 1 interferons that inhibit viral replication, creating antiviral states in our body. As soon as we get infected with the RNA or DNA viruses, various sensory mechanisms or molecules, receptor structures such as toll-like receptors or RAG, retinoic acid receptor molecule, a family of intracellular pathogen sensing antiviral pattern recognizing receptors that we have in our body, they recognize viral antigens such as DNA and RNA structures of the virus. And that will trigger virus infected cells to generate interferon and alpha and beta, and that's essentially shown down below. These interferon alpha and beta then will be released, and those released interferon alpha and beta will be captured by the adjoining cell types, which is hitherto uninfected, and they will become viral resistance. Subsequently, interferons can also trigger CD4s and CD8 T cells that goes into the adaptive immune compartment. With any virus infection, including SARS-CoV-2, we trigger primarily a huge amount of antibody responses that I have shown before. Along with B-cell derived antibody responses, we will also trigger T-cell activation. B-cell activation mediated antibodies will bind to the viral, will block the viral entry into the host cells in us. The goal of the current vaccine is to trigger a very high degree, high level and long lasting antibody responses. Along with B-cell activation, we will also have CD4 T helper cells activation and cytotoxic T lymphocyte activation. These CD8 CTLs and the NK cell, natural killer cell in the innate immune compartment, together, they are involved in the killing of the virus infected cells, thus clearing up the viral reservoir in our body. That's how we overall respond to any virus infection, including current COVID-19 infection. This slide essentially summarizes the viral entry in our body and immune response generation and clearances of the virus infection in our body. It shows this spike protein of this SARS-CoV-2 binding to the S2 receptors. Then the virus gains entry, virus replicates, and when virus replicates, then various cell types, white blood cells, they act as an antigen presenting cells, such as particularly dendritic cells. They will pick up this dead viral particle, and some of them will phagocytose this virus, they will kill the virus inside them and present the viral particulate matter, such as S protein specific peptides, to trigger T helper cells and cytotoxic T lymphocyte activation. On the other hand, B lymphocyte can directly bind to the viral antigen structures when the virus infected a particular cell types or phagocytosed by an antigen presenting cell. Then the B cell becomes activated and triggers anti-coronavirus antibody responses. Once this infection is cleared through natural immune responses, what remains in our body is memory B cells and memory T cells and memory plasma cells. And that is another goal for current vaccine development, that through the vaccination, we want not only to trigger a very high degree of immunity, B cell responses, antibody responses, and to some degree T cell responses, but we should also be able to develop a memory B cells, memory T cell responses in us. And that should live in our system for a very long period of time to protect us against the future infection. And here is the course of antiviral immune responses that is shown on the left-hand side diagram. Forgive me for these broken lines. I was trying to assemble this diagram, but it just didn't align. But essentially, very early on in the infection, as I have said, type 1 interferons, interferon alpha and beta, along with NK cell, natural killer cell activation occurs. They are very essential in defeating virus infection very early on, within days, within the first 24 hours of viral particle infection in our body. Then as the days goes by, within 5 days, we will have antibody responses. This blue line shows the antibody responses. And prior to that, we will start generating T lymphocyte responses, particularly cytotoxic T lymphocyte responses. And about in day 12 or so, through the activation of these antibody CTLs and very early on NK cell and interferon responses, we will control the virus infection and viral replication in our body, and we will clear up the viral infection in our system. And the right-hand panel shows SARS-specific infection and T cell activation, antibody development, and thus taken together, all this adaptive immunity will clear up the viral load in the infected individuals. What will remain in the patient's body is surviving T lymphocytes and antibody populations and memory T cells and memory B cell population to fight infection or keep us protected against future infections. And that's the goal for vaccine development. So with that background of how the infection and immune responses and pathophysiology, pathogenicity of this infection, let's look into very briefly, how do we detect this infection? There are 3 different types of tests at this point available in the market. The primary detection is RT-PCR in real time that detects SARS-CoV-2 RNA sequences. This is a diagnostic, very early disease detection protocol, and it can be also used for contact tracing and isolation of the infected individuals. Subsequent to RT-PCR type of test development, antibody testing came into the market that essentially detects virus infection-derived antibody responses. Previously, I have shown that within 5 days, IgM, and subsequent to that, by days 10, we have IgG class of antibody found in the circulation of an infected individual. So at that point, if we take a little blood sample and send it to the lab, we can detect spike protein specific and other structural protein specific antibody responses in the infected individual. So therefore, antibody detection can be utilized as a detection tool in the later phase of infection and also for surveillance and monitoring, meaning if we go for an antibody serosurveillance in a nationwide or a zone specific, then we can establish the fact that either these individuals were immunized, vaccinated, they generated antibody, or they have been naturally infected, came out of the infection, cleared up the infection, and now they have the circulating antibody in them. Very recently developed rapid antigen test, RAT, also detects SARS-CoV-2 antigen and also antibody presence in the serum of the individual. These are immunofluorescence-based. Qualitative detection can be done at point of care situation, meaning at the clinic, or if at a football game or a cricket game, at the point of entry, individuals can come in about 15 to 30 minutes earlier. They can take a rapid antigen testing, and within 15 minutes, we will learn the results of this antigen testing, whether they are carrying the viral particle and antigenic material in them, or even antibodies can be detected through this testing. But there is a little bit of a drawback about this mass usage of this testing and solely relying upon this rapid antigen testing. About up to 15% can come up with a false positive results. But nonetheless, these are very, very good tests, very specific tests. They can be used at field, large screening, even at the airport. Various airlines for international or long distance traveling, they are thinking of adopting this rapid antigen testing at the gate so that we do not have to show real-time RT-PCR within last 72 hours upon boarding the flights. There are a couple of different versions. Sherlock Holmes, this is a detective in the Western world and failed the test. Another detective in Indian subcontinent, very well known. CRISPR-9, Cas9 technology was used to develop these tests. So all these tests are currently available, and in combination of these tests will help us not only detection, but monitoring and surveillance of this infection. Now let's get into the vaccines. These are the molecules critical for detection and vaccine development. What I'm showing here is spike proteins diagrammatic representations. This is the so-called crown jewel of this infection in a negative way, because this establishes the infection, and this has various domain, receptor binding domain, S1 protein is a trimeric structure, S1 and S2, and various other domain. This sticks out of the viral membrane, and this is the anchoring part. Now, we have understood, scientists and clinicians have understood the spike protein structures in detailed sequencing, amino acid organizations, and etc. And through understanding of these spike proteins, now came the vaccines. There have been many thoughts how we can create a vaccine, again, utilizing spike protein structures and various therapeutics, such as receptor binding domains, domain-specific nanoparticles development as a therapeutics, meaning if we can create a decoy structure mimicking the receptor binding domain of this SARS-CoV-2 virus infection, then essentially that can create a steric hindrance in an infected individuals, when that will perhaps bind to the S2 receptor structures. And one can think of creating a serial vaccines where the core molecule will be a bioparticle, and on top of that nanobioparticle, we can insert various spike proteins and structural proteins of this virus. And these are the various ways scientists visualize that how we can go about creating a vaccines very quickly and maybe lead to the development of therapeutics. And those efforts in last 1 year led to the development of various currently available vaccines in the world. I'll take a moment here to really impress upon and understand what really happened in last 1 year. I'm a viral vaccine researcher. Actually, I started my PhD work in India working on a vaccine development project on snake venom poisoning. So for 30 years, I have worked on vaccine development research areas. I understand how tedious and difficult it is to come up with a vaccine, bring to the research level up to the regulatory affairs and into the market. It usually takes about 15 years, 10 to 15 years from the inception of the preclinical analysis of a vaccine molecule and through phase 1, phase 2, and phase 3 clinical trials, completion of those, and product development, product manufacturing, and approval processes brings the product into the market through safety and efficacy approval. But luckily, All these things have been accomplished in 1 year, just in 1 year. At the very beginning of 2020, when the genetic sequence of SARS-CoV-2 was released by the Chinese scientists, it's a serendipitous development. The scientist who was traveling from China to South Korea or somewhere else, perhaps, from the airport, he released the sequences into the gene bank, accessible worldwide, freely available to every researcher in the world. And he was in talks with his collaborators in South Korea. Should I release the sequence? South Korean collaborator says that, yes, go ahead and release the sequence. So from the airport, from his cell phone, this Chinese scientist released and deposited the gene sequence of spike protein. And that was the beginning. That was happening early in 2020 by March or January through March in some time point. Then through 2020, scientists, clinicians worked at a breakneck speed worldwide to develop the COVID-19 vaccines that we have currently not only approved, but available in the market. Following the tremendous and heroic efforts from the scientific and clinical community all over the world, currently we have viral vaccines, which are inactivated weekend virus-specific nucleic acid, DNA, RNA vaccines, viral vector-specific vaccines, and protein-based vaccines in all 4 categories. Early on, these were the numbers of individual category of vaccine platforms tried all over the world. And it is a success story. There is much to celebrate. How quickly scientists and clinicians not only went about visualizing, imagining of a vaccine, but they actually created one. And that is available. It's a success story. We need to actually recognize this aspect. And it's a tremendous collaboration worldwide that led us to the development of these various vaccines that are currently available. Mostly targeting spike proteins, wild spike proteins, be it a genetic materials or the protein antigen, because spike proteins binds to the S2 receptor, setting up the infection. So the thought is that if we can inhibit that viral particle binding to the S2 receptor, perhaps that could be a good first generation of vaccine. And that eventually came to be the true, scientifically true, because all the vaccines that we have currently are good vaccines, and they're all targeting the spike protein, either mRNA sequences or DNA sequence inserted into adenoviral vector, or simply spike protein as such, coming up with Novavax formulation. And this diagram shows that how scientists originally visualized development of these vaccines. So be it a spike protein gene sequences or viral spike protein itself, they can be injected into the animal and subsequently to the human being. And that will be picked up by antigen presenting cells, and that will trigger antigen specific spike protein, antigen specific immune responses, and keeping us safe. And here is a viral particle, virus-like particle that yet has to come into the market, but people are trying to come up with a viral virus-like particulate vaccine development approach. Taken together, these are the vaccines currently available in the market, and many are in the works, should be coming out shortly worldwide, many parts of the world. So what are these? Primarily, we have mRNA-based vaccines very early on released by Pfizer, BioNTech, and Mrna that targeted S protein-specific mRNA sequences formulated in 4 different types of lipid formulations. After that, or around that time, we had AstraZeneca's adenoviral vector. Adenovirus is a virus. Most of us are carrier of adenovirus, and AstraZeneca utilized a chimpanzee-specific adenovirus, AD26, to insert spike protein-specific DNA sequences in them. This is from Sarah Gilbert's lab at the Oxford University, who studied adenoviral vaccines and not adenoviral vaccines, SARS-CoV-2 infection and vaccine development against MERS and SARS for the last 15, 20 years. This is the formulation picked up by the AstraZeneca, now currently available worldwide. Then came with Johnson & Johnson vaccine, who also utilizes adenoviral vector, but they utilize human adenoviral inserted with a spike protein specific DNA sequences, and Gamaleya and CanSino Biologics. These are the Chinese companies. They also are involved in this particular vaccine development technology. Gamaleya is perhaps a Russian company. Subsequent to that, various other companies also picked up a DNA vaccine platform, IonoVio. They are working, not much information currently available on IonoVio's vaccine development, where they are now, but very interesting information and good news about Novavax, another US company who solely targeted spike protein as protein expressed in the expression system. And they utilize the spike protein formulating with this particularly proprietary adjuvant, and that is triggering a very good immune response. Soon it will be submitted to the FDA. Actually, it had been submitted last month for preliminary approval, and look at, I have some friends who are at the FDA, CBER, who had these files, and in a private communication with them, they asked for additional information on Novavax vaccine. But as of now, the information released by the company is very, very good. It is a traditional protein-based vaccine that is triggering about 90% protection against infection. But shortly, it will receive regulatory approval in US, and in India, they are in talks with, I think, Serum Institute, but I could be wrong about that, with a vaccine manufacturer. They will also manufacture Novavax vaccine in India to distribute in Indian community and worldwide. Another promising protein-specific vaccine is Pitcovac. University of Pittsburgh's vaccine development researchers are working on it. They are coming up with a transdermal patch formulation, meaning it will not be an injectable formulation, but a patch formulation vaccine will be developed through that effort. Killed virus vaccine, leading the pack is Covaxin formulation from Bharat Biotech, India, and Sinopharm from China. These are good vaccines, but unfortunately, not much clinical information is available on this Chinese, Russian, or India-specific Covaxin formulation. They are good, but they are saying that it's good and you trust us. Very, very scant clinical information, efficacy, safety informations have been provided by the respective regulatory bodies and the vaccine developer and manufacturers. And right-hand side, again, you see the how corona vaccines will work, be it a DNA, RNA sequences or protein vaccines. When injected into the individual in the arm, they will trigger antibody responses and T-cell responses. And with antibody responses, they will block the virus infection in an uninfected individual when that individual is immunized with these viral formulations, viral vaccine formulations. A little bit of efficacy data, safety and efficacy data. These are all very safe vaccine formulations because as these vaccine formulations received emergency use authorization, EUA, meaning they are not regularly authorized drugs. Yes, vaccines are drugs because the definition of drug is a molecule that can trigger change in an individual who receives that treatment or injection. Therefore, these vaccines all received emergency use authorization, EUA, from various regulatory bodies because they did not or could not come up with a very lengthy clinical trial and safety and efficacy. But that should not convolute our thought process about the safety of these vaccines. They have been tested on thousands, sometimes 30, 40,000 individuals. They are the vaccine clinical trial individuals who were enrolled in phase 1 through phase 3 clinical trials, and only after safety and efficacy in those individuals have been studied, and that results were reviewed by the panels, by FDA, by CBER, and worldwide review panel. I sat on the CBA review panels when they were reviewing mRNA vaccines for Pfizer and Moderna because it was available, open to everyone. Now I have seen through that process how rigorous was that review, and through the questioning, through the looking from the legal, ethical aspects, bioethicists, clinicians, vaccine researchers. These are the independent panel. 23 of them came throughout the country with different expertise, different background, and they identified, they dissected every aspect of safety and efficacy of these submitted data on these vaccines. Only after that, the vaccines received approval by CDC, World Health, and they were allowed to be utilized, vaccinated in the individuals worldwide. So we should not have any doubt about the safety and efficacy of this vaccine formulation. And what are the efficacy? 95%, 94% from Pfizer, BioNTech, and Moderna vaccines, Mrna vaccines, very high degree of efficacy. These are two shot, 21 days or 28 days apart, whereas Johnson & Johnson vaccine shows somewhat less of efficacy, around 70% efficaciousness with one shot, whereas AstraZeneca vaccine, another adenoviral vector, comes up with two shots, 28 days apart. They messed up in their vaccine clinical trial, data analysis, and providing the data not only to the regulatory bodies, but also to the public release. But it's a good vaccine. Overall, 60 to 90% efficacy and average about 70% efficacy against original strains and some of the variants, which I will show the data subsequently. Sputnik V, Russian vaccine, they claim that 95% efficacious, two-dose vaccine 28 days apart, but really no clinical data is available. So is Bharat Biotech's. 2 doses, killed virus vaccine, 78% protection. That's a one-line clinical data I could find searching the literature. Sinopharm, 2 doses, Chinese vaccine, 21 day apart, 79% protection, and it prevents infection and hospitalization at this rate. And as I talked about Novavax, which is not yet approved, it's a protein vaccine, traditional protein vaccine against other viruses, such as many other viral infection-specific vaccines we have that comes with a protein vaccine, such as Hep B, Hep C specific vaccines that we have currently in the market. Novavax approached or adopted the traditional route of viral vaccine development, isolating the S protein antigens and formulating with a proprietary adjuvants. Their efficacy rate is very, very high, about 90%. Very shortly, this vaccine will come out into the market, hopefully. And right-hand panel shows the various vaccine formulation, their maintenance, and various doses, and a little bit of cost, how much they cost. How these vaccines compare? Well, they're all good vaccines. And all these numbers that we have seen in the previous slide, on this slide, are the calculated numbers of protection, meaning these vaccines, when they went, these vaccine manufacturers and developers, when they went about into the clinical trial, they calculated how many individuals got infection in the vaccinated versus unvaccinated individuals' infection. And through that statistical calculation, this protection information came out into the market. So that's about the vaccine efficacy. Now, these are the vaccine availability globally, and AstraZeneca vaccine formulation followed by Pfizer, Moderna leads the pack, and subsequently Sinopharm, Sputnik, and Covaxin, Bharat Biotech. And these numbers are the countries where they are being given, and these are expanding. And this is the summary slide for various vaccines that are currently available and would be available worldwide, such as AstraZeneca and Novavax. And this is a summary slide. And this medical letter freely available. One can look into the medical letter for not only the vaccine information, but various treatment protocols and etc. in the context of COVID-19. Yes, it did induce immunity, both all Pfizer, Moderna, and all the vaccines. And moving forward with the adult individuals, now Pfizer and Moderna vaccines are authorized for pregnant, lactating women, and vaccines for vaccination for 12-year-old and over, meaning school children can receive Pfizer vaccine. They're also currently doing short, small clinical trial in a dose, means in age-specific 12 through 9, 9 through 6, 6 through 3, and up to 6 months of age babies with Pfizer and both Moderna vaccines. That will shortly come out into the market with the approval for immunization to 6 months of age and upwards. And also Moderna just completed a gamma variant specific vaccine clinical trial in South Africa. That is the local variant, and it has been very promising in the context of clinical data. Immune correlates of vaccine protections, it all triggers T cell and B cell responses, and taken together, those immune responses, these are the prominent real world vaccine protection. Now, these are not clinical data, clinical trial data. These are real world data, meaning individuals in these countries immunized with these respective vaccines. And these are the real world data, how they have been protected upon receiving either complete 2-dose vaccines or a single dose vaccine as applicable. And collectively taken together, it's all very highly effective and protective, such as in Israel, that has the 61% total vaccination countrywide, highest immunized country in the world, that shows 95%, 96% protection. In Kentucky, and against this B117, this Delta variant, the original vaccine is showing 61% total protection. I'm sorry, 61% total vaccination, and also it's showing protections, high degree of protection against this Delta variant. And these are the infection protection against symptomatic infection, also in transplant and immunodeficient patient population, meaning who are originally very vulnerable to infection. These vaccines are also triggering high level of immune protection in them. In Kentucky, in US, 86% protection. Mayo Clinic, they analyzed about 40,000 individuals who are going for surgery through their clinics, and it has shown high degree of efficacy, 80% lower risk of testing positive for COVID-19, and full vaccination triggered 97% protection. Very recent data from CMC Vellore in Christian Medical College in India, they looked at about 7,000 of their workers. When they received 2 doses of the Covaxin, they were protected against 65% against infection and 77% protection against hospitalization, etc., compared to the non-immunized individuals. So collectively, this data shows that these vaccines are highly protective. In last few minutes, we will examine the variants and vaccine protection. These are all the variants that we have talked: alpha, gamma, beta, and delta variant. Now, although these variants generated in individual countries, yet they are now found all over the world, including US. Particularly this UK alpha variant and delta variant are causing concerns in various parts of the world because they are little more transmissible compared to the original strains of the SARS-CoV-2 infection. But good news is that although these variants are highly infective and transmissible compared to the original strain, yet the original strain-specific vaccines are highly effective in protecting the infection in them. For example, a recent study found that B117 making up to 58% infections, although there is no association of the severity of the infection with the transmission. And current vaccines have been found very ineffective against even protection against B117 variants. That is the most rapidly circulating variant in US and all over the world. Very recently, Moderna developed Mrna vaccine with a particular sequence for South African variant specific sequence B1315, and that short clinical trial by Moderna in South Africa shown very promising results that this is their original vaccine. When they are added with these variant sequences specific for South African variant in a 50-50 ratio, they have been found to be highly protective. And here is the protection data: 88% protection with Moderna Pfizer vaccines, also about 88% protection by AstraZeneca vaccine. J&J vaccine also did very well against the gamma variant. And these are the Delta variant specific protection data. Although we do not have a J&J vaccine protection against data against Delta variant, but because AstraZeneca and J&J vaccine utilizes the same platform and technology. It's expected that J&J vaccine will be as protective as of AstraZeneca vaccine. So these are real world data, and beta and gamma variants required actually in a lab setup research showed that about 3.5 to 10 times more antibody for neutralization of the viral particle, regardless of the origin of the vaccination, infection-induced or vaccination-induced antibody responses, or the monoclonal antibody drugs that are currently available in the market. Moving forward, a very, very recent thought process is that can we come up with a superimposed vaccine such as mRNA vaccine, then we come up with a adenoviral vector vaccine such as AstraZeneca or J&J vaccine. Maybe that will trigger a long-term immune response, immune memory production. And also these mRNA vaccines, as I said, that can be utilized with J&J or AstraZeneca vaccine. It doesn't need to be stuck with one vaccine formulation. It may work well, although no clinical data is yet available. Here is the breakthrough reports, meaning in the infected individuals, there has been some breakthrough, meaning these individuals have been vaccinated, yet they contracted infection. Because no vaccines are 100% effective in preventing illness, but still, all these vaccines are highly, highly protective against prevention of infection, particularly hospitalization, death, and moderate through severely infections. But we have to keep in mind that there will be some breakthrough infections. What's the data? Out of 87 million people fully vaccinated, only 7,000 or little over 7,000 got breakthrough infection. Now, with this data, you can understand the efficacy of these vaccines. Adverse reactions of these vaccines. Yes, they triggered adverse reactions such as local adverse reaction, pain, warmth, tenderness, and etc. Blood clot has been observed. Unfortunately, with AstraZeneca vaccines that had been stopped many times till scientists and clinicians understood these adverse reactions, but that's the frequency of 1 in 100,000 individuals, or about 18 deaths, about 34 million vaccinated individuals. Now, that number of people unfortunately died who received immunization. But one can argue that if those individuals, 34 million individuals who are unvaccinated, one can observe some clotting cascade related illness and unfortunate death, but no death is desirable, particularly in the context of vaccinated individual. But we have to keep this data in the context. Therefore, correlation is not causation. And 3 to 5 cases of severe allergic reactions in about 1 million of mRNA doses. And here's the data that shows the serious side effect and side effects through vaccination in this age group, 11 in a million or 4 in a million, but relative information or data due to dying out of the infection, 23 or 800 in a million due to coronavirus infection, and dying due to accident injury is way higher, and dying due to road accidents in this age group, or even hit by a lightning. So we have to keep this adverse reaction in the proper perspective when we talk about the data, when we bring the patient confidence in improving the rate of vaccines. Finally, last few minutes I will take back to the normalcy. I will utilize India's current debacle in the context of COVID-19 infection without going into the details of the data. What is the lessons we have learned in the infection and vaccination in India, and how collectively we can move forward taking a lesson, what happened in India, and they're still coping with it, perhaps at the very tail end. Now, the infection was increasing, and in April, this is the rate of infection and the rate of vaccination, although the vaccines were introduced very early on in January through February and March in India. And this variant that are surging in India, new variant is here, but the original strain was infecting. These are the numbers. And collectively taken together, all these data shows, and this is the latest data, I found it yesterday as of 28th June, as of a few days back, UK versus US versus India. Although the numbers, total vaccination in India is about this, and compared to the UK and US vaccination number is pretty similar in number, but we have to keep in mind vaccinated is only 4.2% of total Indian population vaccinated, whereas in US, we have about 50% of the US population fully vaccinated. That's where the exact explanation of the data set comes into the play. Because if we just tell that India has immunized same number of individuals as of US, we are not doing justice to the populations of India, because India has 10 times more population, at least compared to the US population. Therefore, when same number of individuals immunized in India triggers 4.2% immunization countrywide, same number of individuals immunized in US triggers about 50% immunization. With those data analysis, there is a tale of vaccine inequality and vaccine availability throughout the world. That's also in concern. And this is the total vaccines per 100 people worldwide, different countries. Leading the chart, Israel, and followed by various small countries, and India, abysmally low level of immunization at 6 per vaccine doses per 100 people, individuals. Overordering of vaccines, yes, it has been observed that many affluent countries, Western worlds, pre-purchase these vaccines from various manufacturers such as US, Canada, UK, European Union, Australia, etc. African Union also stockpiled vaccines. India abysmally low at 4%. Now, this over stockpile of vaccines by various countries in the world needed to be given to the countries in the world where there is short supply of the vaccine. This is the data I could gather as of yesterday from Mauritius. Specific data. Confirmed cases is about 1,800 deaths, 18 early on. Dr. Mohadev informed me that as of today, 22 deaths in the country. And this is the vaccine tracker data, doses administered, people fully vaccinated. These are the numbers. Percentage of people fully vaccinated is over 17%, close to 18%, which is good, but it needs to be improved. Finally, I will leave the audience with some parting thoughts. We have learned quite a bit, but much needed to be learned. We need comprehensive testing, treatment, and nationwide, worldwide vaccination guideline. We need to have a centralized, uniform public health policy. We need to develop vaccine confidence, overcome hesitancy. We need to take care of the vaccine inequality in the world. Policies to mitigate social and economic disparity in infection and mortality, coming up with quick testing, tracing, quarantining, and advocating mask wearing and social physical distancing, if necessary, facility closures. Finally, will there be universal combined vaccines? Yes. Clinicians and scientists are moving towards that. And final thought is that will vaccination give back our old life back as it used to be pre-COVID era? Yes, but it will be gradual in phases. We still need to quarantine, surveillance testing, genomic sequencing, monitoring, tracing the variants. Isolation and quarantining and contact tracing will mitigate the spreading of these variants. We need to follow public health guidelines overall in the country and for containment and the mitigation strategies. Perhaps human behavior will do more than vaccination, and will determine moving forward how soon we can go back to the normal. Overall, the states and the countries are diverging against these guidelines. And COVID-19 moving forward may remain as a community-based infection unless there is no magic number. Perhaps 70% population in a country and in the world are vaccinated, developing herd immunity. We really cannot declare a victory, but it will come in a zonal, in a country-specific manner. Access to testing, genomic sequencing testing will help us getting back to the normalcy, but will depend greatly where one lives, meaning it will be country-specific, slowly worldwide episode. Thank you for your attendance and your attention, and I will entertain any question. And I dedicate my uncle, sadly, who passed away last night in India. And he was about to attend my seminar. He was a physician. It's my dedication to my uncle. Thank you very much.
Thank you. Thank you so much, Dr. Mandal, and very sorry to hear about your uncle passing away because of this. It was a very nice presentation, very clear information which we received about different vaccines, and I can see there are a lot of questions which are popping up because yes, in the general public's mind, there somewhere there is, as Dr. Mohadev in the opening remark he was mentioning about, there is a question and a confusion related to the vaccines and different variants, how we are going to look into that. So there are a couple of questions. I think we have some more time, and I request the coordinator of the webinar, Dr. Khyati Modgil, to come up with some of the questions which will be put to you, and if any other panelists also would like to join hands to answer that. Dr. Khyati, over to you.
Thank you, Dr. Wadhwani.
Yeah,
First, can you hear me, right?
Yes, I do hear you, and I also see the chat box. Go ahead.
Yes,
yes.
Dr. Mandal, we just have a request. Can you unshare your screen so that we can?
Sure. Give me a second. Stop sharing. It's done.
Thank you so much. Go ahead, please.
So a very good evening, everyone. First of all, very good morning, Dr. Mandal. I thank you for providing the insight about the various vaccines, its comparison, efficacy, and the variants. And it was a very informative session. I would say it's a comprehensive and very rich lecture about the COVID-19 and its all variants, and which I hope that it would have cleared the doubts of everyone among the audience and the participants. And now the session is open for QA, question answer. I request the participants to put their questions in the QA or the chat box. But in meanwhile, we would like to take some questions. Dr. Mandal, there are some interesting questions. First question is, like, every time, nowadays, like COVID rate is increasing, whether there is any need of third dose for the COVID?
Right. The very good question and very good insight. As of yesterday, there is already a talk started in UK, in Britain. In their clinician and their scientists, they are thinking of coming up with a booster dose. Actually, it has already started a few months back in US. Both Mrna, Pfizer, and few other vaccine developer, AstraZeneca, are already in the process of developing a booster dose, utilizing these variants of concern specific sequences to come up with a booster dose, perhaps by the fall. What I wanted to mention about this booster dose, third shot or a second shot, as it pertains to the particular vaccine formulation. If it's a Moderna, Pfizer, or AstraZeneca, it will be a third dose, booster dose. Or if it's a J&J vaccine, it will be a second dose booster. In UK, there is already a talk which they started discussing that by as early as by September, they might introduce a booster dose to keep the already immunized individuals protected against these emergent variants. towards that, I provided a very snapshot of very early data, the clinical trial done by the Mrna vaccine company in the South African context, targeting the South African variants. Where they did there is that they utilized a vaccine booster dose, where 50% of the booster dose was from the original mRNA sequence, and rest 50% came targeting the South African variant-specific mRNA sequences of spike protein. And when they combined that vaccine formulation 50-50 from both ends, and they did a short clinical trial with a limited number of individuals, they actually saw a very good protection in those booster dose vaccination in those. So it's already in works. Short answer is that we may as well expect the guidance will come out from regular competent authority worldwide to adopt a booster dose by fall through winter 2021. That will not only boost up our current existing antibody and T-cell responses towards the original sequence, original vaccine, it will increase the longevity of those antibody populations that we already have in us if we got the immunization. It will also improve our immune condition, immune status, protection level towards those variants. So the answer is yes.
Okay. Thank you, sir. Coming towards the next question, now there is always dilemma, like what kind of mask, is important,
yes,
and what kind of mask will prevent, can prevent the prevention of this pandemic.
Yes, sure. I used to have a science article that was published last year, early on in 2020 by March, that did a phenomenal experiment utilizing a simple cell phone and a laser beam. And they asked individuals to wear different types of masks, bandana, handkerchief, and etc. What they simply did, they asked people to cough up, and they tracked that aerosolized particles that are released from the mouth of the individual that's coughing, going through that laser beam and through the cell phone, utilizing a particular software. From their cell phone, they captured that particulate matter going through the aerosolized beam and capturing the image. And they analyzed using algorithms, statistical algorithm and computation. What they found is that N95 is the best mask that can protect us against aerosolized particulate matter inhalation and infection. Second is KN95 mask, little below N95, because N95 mask needed to be fitted, needed to be practiced. That takes some time. KN95 mask, which is right below the N95 mask, that protects this viral particle transmission over 70% level. And that over 70% viral particle transmission barrier data I have utilized in one of my slides. Mask wearing prevents protection. Next comes surgical disposable mask, that triple layer. That's also highly protective. But really, the bandana, the handkerchief, or any other cloth masks do not really provide any protection. So with the emergence of these variants, which are highly transmissible, because their S protein structures have been mutated so that they become more sticky to the S2 receptor structures in our body. Therefore, they are highly transmissible compared to the original strain. We will do better if we really want to wear a cloth mask, then double up the layer with a surgical mask, or even better, if we can wear a surgical mask, disposable surgical mask, and top up with a N95 type of mask or KN95 mask. That's the best protection. Through clinical data and lab research have shown that wearing a mask essentially can cut down the transmission up to 70% level, meaning if we are in close contact with another infected individual, and if I am wearing a mask, double layering, I can prevent transmission up to 70% level in me. So that's the data we have currently. So yes, we should wear any of these masks to facilitate prevention of the infection in us.
Yes, because recently we were studying about and we read about the WHO says about the double mask combo. also.
Yes.
That is highly preventable against
Highly
this
preventable,
pandemic.
yes.
So
that's true. And the current guidelines for CDC, and is my personal practice. I have been vaccinated by January as I was working in the immunization clinic with our university. But I still go out. So US came out of the mask mandate. It's over in US. Although the variant infection are increasing in the pockets. Still, when I go out in the grocery, when I go to the gym, in close contact with the people that I do not know whether they are vaccinated or immunized, I wear a mask. It gives me a sense of protection, heightened immunity, not only immune, means heightened protection. One comes from the immune responses that I had through immunization, one's creating a physical barrier just over my mouth and my nose. So I would highly suggest even someone got their immunization, still do wear a mask when you go out in the community, in the grocery, in the contact of students, etc. That will create a second barrier to protect us against the infection.
Thank you, sir. Moving towards the next interesting question is, are there any nasal vaccines in pipeline for this pandemic?
There has been talks, have not seen data, but the basis for nasal vaccine, viral vaccine, comes from the one viral vaccine against influenza that's called FluMist. FluMist is a viral vaccine that had been developed by Professor Hunayn Masab at University of Michigan College of Public Health. When I was at University of Michigan, I had close contact with him. He was a pioneer researcher who fundamentally discovered that many of these viruses do carry genes in them which are thermosensitive, meaning the core temperature of our body is higher than the surface temperature. So he utilized that fundamental observation that when we get infected with a viral infection, viral particle through our nasal or through our mouth, when it goes inside the lung to set up the infection, lung temperature is slightly higher than the surface temperature of our body. So he manipulated some of the thermosensitive gene in influenza virus and created a FluMist type of live attenuated influenza virus vaccine that is already in the market for last about a decade or little over a decade, actually about 20 years in US. It triggers very good mucosal immunity. The context of this development and approach is that if you think clearly, almost all the virus infections and majority of the infection comes into our body through mucosal exposure from influenza, HIV, SARS-CoV-2, even gut-associated E. coli infection, bacterial infection, essentially is a mucosal infection. Now, if we can trigger a mucosal immunity, such as secretory IgA, a particular fraction or a subpopulation of antibody, that naturally we generate through mucosal immunization and immune responses that covers the mucosal epithelial layers in our body, that will naturally prevent this virus infection also. So there are works in the works, meaning research in the works to come up with a mucosal SARS-CoV-2 specific vaccine formulation following the guideline or following the information that we have against natural seasonal influenza infections, FluMist type of viral vaccines.
Okay, sir, thank you for the clear information about this question. Now, moving towards the next question is like, if the person has taken already the first dose of COVID-19 vaccination, so if he or she is taking the antibiotics, whether it is gonna affect the efficacy of that vaccination?
No, absolutely no. There are many misconceptions that if I'm on anti-inflammatory drug to treat any rheumatoid disease condition or inflammatory disease condition, or if I'm on antibiotics or any other medications, it might compromise the vaccine efficacy. Upon immunization or natural infection, there are certain cell types that gets involved that crosstalks with each other. What are these cell types? These are the dendritic cells, other antigen presenting cells, part of the white blood cell family in our body. They will pick up the vaccine antigen through natural infection or immunization. Then they will trigger the educate T cell, B cell activation. They will generate antibody responses, cytokine responses, and etc. Now, unless these drugs, specific or any drug, targets specifically to those white blood cell populations, and that too, those are involved in the immune activation towards vaccination or natural infection, other medication should not compromise immunization-induced immune responses, antibody responses, or infection-induced immune responses. So there should not be any doubt that you need to stop your current medication so that you can have a better immunity when you are getting your shots. No, it is not. And moving forward, I can give you a very clear clinical information. Many people who has heart disease, who has high blood pressure, they take H2 receptor targeting drugs and herbs, right? As a pharmacist, you know that.
Yes.
They are not advised not to stop those medications in a thought process that because they also target H2 receptor structures in our body, in the biochemical pathway for those drugs, pharmacologically, they should not stop taking those drugs. There have been clinical trials in the individuals who were taking those drugs, where are the infection less or more in them? Answer is no. Those drugs should not be stopped in those individuals, and no medication should be stopped to enhance the immunity or increase the immune response through natural infections.
Okay, sir. Now we will take just last two questions. And the another question is, do we need to change or modify the testing protocols? Are there any need for that?
Testing protocols is a very important question, a very interesting question, because testing protocols needed to be modified dependent on the particular strain of infection variants. Yes, genomic sequencing. To answer straightforward, coming to your question, is that along with RT-PCR, if possible, we should do more genomic sequences. INSACOG is such an organization, works in India, and very country-specific genomic sequencing facilities should be involved into the community genomic sequencing and etc. One example is that a particular approach has been adopted, not only tracing, but sequencing from this virus from the sewer water, meaning this virus ultimately travels down through our bowels and end up in our sewer water in a community. And worldwide, there is a protocol in place. They are not only tracing the virus, but using the sewerage water-specific viral particle, they are doing sequencing. By doing so, at a glance, we can understand, we can see an overall picture that in this region, even in my town, in Las Vegas, greater Las Vegas Valley, there are emergences of Delta variant, particularly in that area, sewer water carrying this through genomic sequences. I can identify that area individuals are triggering or infected this Delta variant specific infection. So genomic sequencing should be taken in place in parallel with RT-PCR specific detection. Along with that, I would mention that rapid antigen testing can be very effectively deployed, bringing the school children, the students back into the campus, because it's very, very lowly priced, it's very affordable, and it can be done without usage or needing of any instruments or any gadgets, and test results can be found within 15 minutes. So we need to modify depending on where the testing is given, what type of information we need to get, and what are the availability of the testings or the kits in the community.
Yes, sir. Thank you for that. And coming to the last question, what is your say on Delta variant, which is now in the limelight completely?
It's $1 million question. I wish I had a crystal ball to answer that question. Delta variant is of concern worldwide. We have seen the debacle in India. we are waiting to see something serious might as well happen in the southern states of US where the immunization rate is low. So taking these two observations together, what I will say, although Delta variant is a variant of concern and might as well trigger high level of infectivity, disease severity, unfortunately mortality, if the treatment protocols and facilities are not available, such as mundane as oxygenation, yet we can stop the spread of Delta variant worldwide if we get our vaccines. Now, in the US, we are in vaccine access zones, meaning if anyone can travel, any of the participants can travel to my hometown, Las Vegas, I can take that individual to get a shot. It's so freely available. That's not the case everywhere in the world, but we should all strive to immunize more people in the community, in the country, to stop the spread of the Delta variant. And moving forward, now it is called Delta Plus, and maybe it will acquire few more mutations. And how we can stop that spread of these dangerous mutants? If we can vaccinate ourselves. If we immunize ourselves within 15 days, we will acquire enough immune protection so that majority of us will be immune to catch this variant. And remember, once again, viruses only mutate when they infect us. This is virus's weapon. It's a tug of war. It's a war. Sometimes we win, sometimes the pathogen wins. Now we have the shield of vaccine that will protect us through this variant emergence and Delta variant. So if we immunize few more individuals and largely throughout the community reaching herd immunity, then we can protect us against this variant along with mask wearing, physical distancing, not going to the parties, not going for wedding type of receptions and etc. That is possible.
Okay. Thank you very much, sir. I think we have covered almost all the important questions. So with the permission of the chair, we would like to conclude the webinar. And on this, I sincerely thank Dr. Mandal for accepting our invitation and delivering this talk, which is absolutely in need of an hour. I also thank the leadership of JSS Academy of Higher Education and Research, Mauritius and Mysore for their constant support and the encouragement. And I thank Dr. Ravindra, Chief Information Officer, for making this online arrangements. And I thank you everyone. And by this, with the permission of the chair, we would like to conclude the webinar.
Thank you, Dr. Kayati. Thank you very much, Dr. Mandal. I think there are many questions which have come up. I think we should have a separate question and answer session on COVID-19 vaccines and variants, because I can see there are around 36 questions, but more or less we thought because of the time constraint, we will take up the important questions. Once again, our deepest condolence to you and your family for your uncle, and thank you very much for joining and giving us the information about the vaccines and different variants. Thank you. I request Dr. Mahadev to conclude.
You are muted, Dr. Mahadev.
You have to unmute it, sir, please.
I would just like to say thank you very much, Dr. Mandal, for this very enlightening lecture. I'm also presenting to you our sincere deepest condolences from JSS Mauritius. Thank you again, and all the best.
Thank you very much. I will take a minute. Thank you, everyone, for the organizer, for your sensitivity. But what I like to mention that although unfortunately perhaps went over the time, not being able to answer all the questions, please forward my contact email address to the respective individuals. I would get back to them with any questions they may have through emails. And thank you once again.
Definitely, I would. Yes.
It was my pleasure, and I hope that we have been collectively being able to reach out a wide audiences worldwide, and collectively this should be a collective effort to defeat this pandemic. We are about there, but we need the last push. Perhaps this type of events would facilitate that last push to defeat this pandemic. Thank you very much once again. Bye, everyone.
Thank you. Thank you, sir. Thank you. Thank you, participants. We are concluding the session, and as Dr. Mandal mentioned, if you have any specific question, you can write it to us, or we will. I have all your email IDs, so I can share the contact of Dr. Mandal, and we can interact on this. Thank you. Thank you, everyone, for joining. Bye-bye.
Bye-bye.
Thank you, everyone. Good night. Thank you.
Good night.